So this is the third day of the training, and we would like to show you the special features of the repository, which are the modules and plugins. Also, how you can use the system on mobile devices, and then at the end I will try to tell you something about the standardizations to develop within Aquaexcel project, and just to point you to the possible cooperation with the Elixir. You probably know what is it. That's the that's the initiative where you can ask for the special services. You can upload the data to some servers, and then they will process the data for you and give it back to you. So I'll just start with a wrap up from the last day. So what we have learned was that I showed you how to again how to create a protocol template, then how you can create a protocol based on the template, and I showed you how you can share and search the data. So these are the main things what you can do with the system daily, because this is what we expect from the system of data management. Today, I will speak about the standardizations. Then I will show you how we can communicate with the measurement devices. And then I will show you how we can process and visualize the data through data processing modules. So I will start with the first thing. So we don't need this one. And I can directly jump to the software. So you know this. So first thing I would like to show you is how you can use the standardizations. It means from the point of view of the terminology. Because if we decide to create new protocol template, I will call it standard. Say okay. Then if you would like to cooperate with someone else and share the protocols and templates with someone else, then we should use the standardized terminology. It means that everyone will understand what the terms, what the names of the different fields here means. So then you minimize the problem with the misunderstanding of your description of experiment. Therefore, several initiatives which collect the standards from different fields of research exist. On the first day of the training, I showed you the links to different standardization portals. So you can see the links here. If you go to one of these portals, then you can search if some standardization exists specifically for your field of research. Of course, it is not all the areas are covered, but some of them exist. For Aquaexcel, during the first project, the standardization was developed, and now I will show you how it can look like. So if you go to the Aquaexcel EU web page, then you can go to menu and see the menu ontologies. If I click on it, then I should have this page. Ontology is the collection of the knowledge which is saved in some special format, and for our needs, the ontology contains the description of the different traits for the livestock. So I directly go to the ontology itself using this link and switch to English. And here we have different ontologies. So what does it mean? I click on Atoll, and the ontology is something like tree, where we have the traits organized in the directories. So you can see that we have different terms for different traits. For example, for the reproduction, we can have very different traits for sex determination. And if you click on every single record, you will see the description of the record. So it means that this tree collects the terminology from the specific area of the livestock. So it could be probably useful if something like this exists, then to use it in our system, because otherwise you will have to learn all the terms and be sure that you are using the correct terminology. So our system provides very simple feature. So if I go back to the system and decide that I would like to describe something that is related to the livestock, then here I needn't to think about what terms should be used, but I can simply ask the system to provide me the terminology. So I click to the terminology button, and right now I have to define the source of the terminology. So I go back to the web page. Oh, sorry, it's not this one. This one. And here we can download the terminology. So I click on that and say download atoll terminology. And what appears is a so-called OVL file, which is the file or the structure how the terminology is described. So it's readable for the computer, not for the humans, as you can see. So I can simply copy the link to this ontology and paste it to our system, Control V, and say just OK. And right now, the system downloads the ontology, the terminology, and we have it. So you can see that we have the same things as we saw on the web page. And if you would like to use the terminology, I just click Use the terminology and say OK. And right now, if we start to define the name of the controls, then, for example, fish, then you can see that the system starts to offer us the terms from the terminology, from the standard. So this is a very simple way how you needn't to learn the terminology, but you can simply start to use it. So if I choose this term, then it will appear in my protocol. So this is the way how the system can use the standardization from different fields. So the OVL files, as I've showed you, is the standard for description of the ontologies. So from the from the portals, which are mentioned here, the files are saved in this format. So it means that the BioWES system can read the standards. So this is the way how we can use the terminology during the definition or design of our protocol templates. So very simple way. Okay, this was related to the terminology, and right now I'll try to show you how we can establish the communication with the measurement devices. So on the sample I showed you from our laboratory where we were with the microscopes and with the cells, the microscope can provide the information about the setting of the microscope. So if we know the structure of the data that the microscope provides, it can be different, of course, for different measurement devices. So if you work, for example, with the mass spectrometry, then the devices usually provide a setting and so on and so on. So we have to know the format of the file. So then what we can create is so-called plugin. So it's very small piece of software which can read the data file that the measurement device produces. And I will show you where the plugins are stored. So I navigate to the installation of the Aquaexcel repository. So I go to my hard drive. I go to the program files, to data partners directory. That's the company who help us to develop the solution, and to BioWES. And here we have the directory with the plugins. So if I open that, then I can see that in my list of plugins, I have two plugins. The first one can read the data from the microscope called BioStation. The second one can read the data from microscope from Olympus. So for specific case, if you have any device, then we can prepare the software for you, the plugin, or we have open interface. So if you are programmers or someone from your department, then you can yourself create the software to read the data. So the plugins are stored here, and if I have some plugins, I can simply start to use that. So for example, for my microscopes, I would like to know when I started the experiment. So for every component, you can see that we have the field, the property external. I didn't explain this one before, but this is exactly where we define how this control should be filled. So if we have a plugin which can read the data you would like to put here from some device, then you can simply click on this button, and in this list here, you should have the list of plugins that are available. So you can see that the only plugin which is available now is this BioStation microscope. So I choose it. And then the plugin offers me the possibilities. What can I read from the microscope automatically? So you can see that the microscope provides information about the magnification, start, end day, number of frames, exposure, and so on and so on. So for our case, I choose a start time and say OK. And now we see that this information can be filled by the plugin BioStation, and we read the start time. So how did it work? Right now I define how the protocol can read the data automatically from the system, from the measurement device, sorry. And later on, so I save the template. Okay, I finalize the template, and now I have it, I can use it. So I go to the list of my templates, and I should be able to see it under standard. And now, if I decide that I would like to measure something, I create new protocol. So I add new protocol, I call it standard, and I have to choose my standard template and say OK. And now I can start to fill the protocol, as you know, but because we decided that we can read the information from the microscope, I can say fill protocol. And what happened is that the plugin I defined during the design of the protocol, so the BioWES plugin, is called. And right now I just have to choose the software or the measurement device I would like to read the data from. So my data are stored here. I just navigate to BioWES. And these files are produced by our microscopes. So here in this file is the information about the setting of the microscope. And I open that. And sorry, the date was not the good example. So I will just try to open this BioStation protocol. Okay, so for this protocol, I defined that the information should be read from the microscope. So what can I do? You can see that it's already filled, but what can I do? I can clear the data. So I say clear, and all data will be deleted, and you can see that everything was deleted here. So You can see that it's empty right now. So this is the way how can you clear your protocol simply. And right now I click on failed protocol. So again, I have to select the file. It's here, and select the file with the information about the setting and say open. And you can see that the information was filled in. So I have the information about the cell line. I have the information about acquisition period, number of frames, resolution, exposure, and I also have the information where I measured my sample, what points of the sample I use for the microscopy. So this is the way how you can automatically fill the information about the setting of the device into the protocol. So it's faster than you retype it manually, and of course it minimizes the the error which can cause during the retyping. So this is the way how you can read the information from your measurement devices. You need the plugin, but it can be created by ourselves. Or if you have some programmer or you are the programmer, then we have the open interface, so you can implement the software yourself. So this is the information about how we can communicate with the measurement devices. Okay, so right now I close this template. I don't want to save it. And right now I would like to show you how we can process the data directly in the system. So once you measured something and you created the protocol and you stored all the information to the system, then we have the data in. And at the beginning of the training, I explained that the system see the data as a black box. So it means that we don't see inside the data. But the way how we can see inside the data are the modules. So you can see what appears here. We didn't see these three icons before because these are the modules for data processing. So each module is a specific software which can read your data. So the module has to know what is the structure of your data, and then we can again, as similarly as the plugin, we can implement the piece of code to process your data. So again, we have the open interface you can use, and you can create your own module for data processing. So to show you what can we do with the data, I will show you some examples. So for example, we have the module for visualization of the data. So I will navigate to the protocol which was created from the first Aquaexcel project. It was a study about the influence of the fish growth based on the different upscaling of the fish tanks. So if I open the protocol, then you can see that it was created by Nofima. And there is an objective described, experimental setup, different things for the tanks. Here you can see the scheme of the upscaling methods. And we have the experimental data, which at this case are the mortalities if the fish was growing in cage, if the fish was growing after we moved them from 11-meter tank to 2-meter tanks, and so on. So probably we would like to see the mortality, how it looks like. So therefore, we have this visualization module. And if I just choose the protocol and click on Show Charts, then the module offers me the data that are available in the protocol. So I choose, for example, 7-meter tank and say OK. And right now, I can choose the type of the graph. So probably I would like to have the line graph and finish. And what I have is the visualization of the cumulative mortality during the experiments. So this is a very simple way how your data can be visualized directly in the system. So it's one example of the module. This is the data visualization. So another type of the module is the data processing. So if I would like to process my data in the system, then I will use another protocol. So this protocol was created by us when we tested the fish monitoring system in Nofima. It is the system which is placed above the tank, and there is a camera which monitor the swimming of the fish. So you have the images of the fish inside the tank. So there are some descriptions of the system, and what we have are the images from the camera. So we had a lot of images with the fish, and what the module does is that the module, I choose the protocol and call the module, and the module read all the data from the protocol and enable me to choose the images I want. So I select all of them, and the module process the images using image processing methods and detect every single fish in the tank and calculate the information about the speed of the fish, about the 3D position in the tank, and about the distribution of the fish inside the tank. So after some initialization, the module try to estimate the background of the tank and then detect the data. So this is the example of specific data processing. So you can see that we have the images. So the module has to know that we work with the images. And then you can implement anything what you want. So if you are using some methods for data processing, then it can be implemented to our system, to the BioWES system, or some softwares support also to create a module from the software and plug in to some other system, for example, to the BioWES system. So if you are using something for your data processing, then it could be probably plugged into our system and you can process the data directly from the system, from your protocol. So very simple way. So what can you see here is the tank. We have the fish here. So the green objects are the detected fish. This is the outlet in the center. And here on this image, you can see the 3D positions of the fish in the tank. So this is what this specific module does. Okay, I'll stop the processing because it will take a long time. And right now it tried to save the trajectories of the fish, so I don't need it. Okay, and I can close the module. So another example of the module could be, for example, the object analysis. So just to demonstrate that we can have very different functionality of the modules, I will again choose the module with the fish images and run object analysis module. And again, select all the data again. And new window in the system is opened. And what can I see here is the image of the fish from different sources. So here I can see the original image. Here I can see the red part, blue and green part of the image. So this software enable the user to see the different parts of the image. I can go through the sequence, so I can see how the fish moves. And what can I do here is that I can manually label the objects here. So what I'll try is that I will try to mark one fish, for example this one, and so I will try to magnify the fish, and just by simple clicking, the software allow me to choose the fish. So I just mark the border of the fish, and I can go to config and do some settings. So you can see that in the menu I defined that I would like to adapt this objects I defined for the next images. So when I go to the next images, I say go to the next, then the system try to adapt the object to the new fish body. Um, um, it's more or less successful. It depends on the data, of course. Um, so this software allow me to mark the object and then calculate the areas of the object and so on. So again, I can have the trajectories of the fish, number of fish, and so on. So very different functionality with this module. Um, so this is the way how we can see inside the data which are stored in our protocols. So if you have some specific software for data processing or analysis, then you can use it directly in the system. So after you will use our open interface, then new module can appear here, and you can simply use it to data processing in the system. So this was related to the modules, and what I would like to show you as the next possibility of the system is the mobile interface of the system. So because the mobile interface is mainly running on the mobile phones or on the tablet. So what can I show you is that I will try to run the system on my phone. So I just test if it works. Okay. So what can you see here is the remote screen of my mobile phone. So I have it here. And so what I will do on my mobile phone, you will see here on the screen. So I'll try to connect to BioWES system. I already prepared icon on my desktop, so you can see it here, and I simply click on that, and it's nothing else than again web interface. So using your browser, sorry, okay, it works right now. Again, okay, I'm connected. So we can see that the address in the browser is the same as we have for our web interface. So it has very similar functionality, but on my mobile phone I can just fill the protocols, or I can modify the protocols. I can't design the protocol templates because it's not very handy on the mobile devices. So the main reason why we have the support for mobile devices is that you can take your phone, you can go to the field or close to the tanks and fill the protocols directly there without the computer. So I'll try to log into the system. I'll use the same credentials as I'm using for the web interface. I'll fill in my password and sign in. And what can you see is the list of the protocols that I already have. So I can open one of them, or I can create a new one. So I click on Add Protocol, and it asks me for the name. So I can just use the name mobile. And I have to choose a template, so it's exactly the same as what we do in the protocol manager on our computer. So I have pre-prepared here the mobile fish tank protocol, and again I could link the protocol to some existing protocol, but it's not necessary right now. And I say add. Okay. And the protocol was created. So what can I do right now? I can define the description, of course, and so on, if I want to finalize the protocol. But if I move to the next part of the protocol, then you can see that I can define the name of the experiment. For example, this is the test. I prepare the person who is doing the test, so it's me. And then I can go to some other part of the protocol. Oh, sorry, let's say data files. The other part is hidden here, so I can go to other tab. What I'm doing, for example, this protocol was prepared to check the tank. So I just choose the date when I do the check. Sorry, it should be today, but it doesn't matter. But maybe we have till April. So today, we can define the number of the tank. So maybe it's tank number 4. I can check what I did. So I check the oxygen, I check the feeder, I check the fish mortality, and then I can decide about the tank state. So I can say, okay, there was some problem. Because there was some problem, I probably would like to take the picture of the problem. So I go to the data files. So you can see that the protocol is designed exactly in the same way as what we see in the manager. So we have different tabs, and the last tab is the data files. And what can I do right now? I can take the picture of the problem. For example, I'll take the picture of myself. Okay, and then I can say upload to the protocol, and it was uploaded. And once I go back to the protocol and I say save, that at this time everything was moved from my mobile device to the repository. So at this time I should have it in my list of my protocols. So we can simply check. I can go back to our software, and if I go to the protocols, then I should see my mobile protocol here. So it was created right now. You can see the time. And if I open the protocol, then, oh, sorry, it's not this one. Okay, I have to refresh. Okay, go to protocols. Okay, and right now we should have the data in. Oh, how come? We have the data file here, but why don't we have the data itself? I will check on my phone. Oh, sorry, it was not filled. I will go again back to the mobile. Sorry, it was lost. So I fill again the information. Number 4, oxygen, feeder, fish mortality. There was some problem, and I will save the protocol. Okay, so it was saved right now, and hopefully right now we will see it in the system. I have to close the protocol, and okay, yeah, and right now we can see the data in. So this is the way how you can use the system in the field. So if someone fill the protocol, fill the data in, then after the person save everything, it's moved to the server. So at that time, someone else can start to work with the data. So this is a very simple way how you can cooperate remotely from someone else if you are in the field. Or, of course, you go to the field, you collect your data, describe what you did, and later on in your office, you can work with the data because they are stored on the server. So this was the example how we can use the system on mobile devices, um, and that's practically all the extended functionality we can use in the system. What we didn't describe during the course is that you can generate your protocols to the PDF file. So if you need to print the protocols, you can simply click on the generate PDF, and the protocol is generated to the PDF form, and then you can do whatever you want with the protocols. Mm. Uh, of course, there are some settings of the protocol itself. So if I'm in some protocol, I can click on the settings, and here I can change the name, I can change the description. Also, I can change the connections of the protocol. So we can see that this protocol is not connected to any protocol. So I can change it. For example, I can connect it to this protocol. And once I say OK, then you change the link between the protocols. So this is the way how you can change the links after you already created the protocol, because during the definition of the protocol, you define how the protocols are connected. So this is the way how you can change it. So what I showed you today was that I showed you how you can use the standardization. So we have a very simple way how we can download some existing standard to the system, and you can simply start to use it for the definition of your terms. Then I show you that the system can cooperate with the measurement devices. So if your measurement device can provide the information about the setting, then during the design of the protocol, you can define what part of the protocol should be read by automatically from the information from the device. And I showed you that we can also use data processing modules, which we have here, for visualization or data processing. So if you need some data processing, you can do it directly in the system. So you needn't to download the data to your computer and play with the data on your computer. You can do it directly in the system. So finally, we also have the support for the mobile devices. So if you need to do something in the field, then you can simply use your mobile phone to fill the protocol. Also, you can upload some data on the mobile phone. You can upload the picture or GPS coordinates, because usually we don't have anything else. And then you can again process the data, because everything is stored to the local repository. So at the end of the training, I would like to show you that during the Aquaexcel project, several different standards and guidelines were developed. Because one of the reasons why we developed this system, Aquaexcel repository, is that we would like to use the standardized form and methods for the data measurements. If you would like to have the overview of the standardizations and methodologies it was developed, you can go to the Aquaexcel webpage, Aquaexcel EU, go to the results, and if you go to the ontologies, then you can see the ontology I showed you. But you can also go to the deliverables, and then you will see all the deliverables that were delivered during the project. And what is listed here are the reports and describe procedures, for example, for the sanitary prescriptions and procedures for transfers and safety standards, or the best practices for the measurement of phenotypes, or some other some other guides for the methods and for the standards. Um, so what we are going to develop during this AquaExcel 2030 project is that we are going to improve AquaExcel repository, the BioWES system. So it will not support just the terminology, but it will also support the standards for data measurements, so the procedures, the standardized procedures. So the idea is that the user can define in the protocol template how the specific trait should be measured, and you will be able to link the method and describe the method in the protocol template. So once some user will use your template to measure something, then he can simply click on the trait and the method which should be used for data measurement will appear. So this will be the way of the standardization of data measurement. So this will be ready next year, and it will be added to the functionality of this BioWES system you installed. So once you install the new version, the support for the methodologies will be also implemented. Another thing which is going to be developed under the AquaExcel 2020 project is the cooperation with the Elixir initiative. So as I already told you, Elixir provide the services for data processing and for data storage. And what will be developed in the AquaExcel project is that we will try to identify the Elixir services which are useful for AquaExcel consortium, for the members, and demonstrate how you can use these services on 3 case studies. So again, the report about this work will be provided. So after 2 years, we will deliver this description of the use cases. And if you will be interested in the cooperation or usage of Elixir services, you can simply follow this guide. And it will show you how to organize your data, where to upload, where to ask for the services, and what kind of services are available. But it will be developed within 2 years of the project. So from the point of view of the system AquaExcel repository, hopefully we showed you how you can use the system for your work, and maybe you will be one of the users of the so-called TNA projects. So at the end, I will just show you, just to explain what is TNA project. AquaExcel Consortium, as the infrastructure project, offer the possibility that you can apply for this TNA project to go to some infrastructure which is under the partners of AquaExcel project, and you can simply ask to realize some experiment. Then, if your TNA project is approved, AquaExcel gives you the money to travel to the infrastructure, to stay there during the experiment. It pay all the costs of the experiment, and you can realize the experiment, for example, in Norway, in Spain, and at any country of the AquaExcel partners. So I will just show you how the application form looks like, because after you apply for the project and you are successful, then we create for you your project description in BioWES system in AquaExcel repository under your account. So once you start your experiments, then you will have the information how you describe the project and you can simply attach your protocols with the experimental data to this description of the experiment. So you can see that here we have the information about the applicant and so on, and what we have in the protocol is a description of the experiment, what is the proposal, what is justification of the proposal, what is the objective, and so on. So everything what you describe for the project, it will appear in the protocol in the Aquaexcel repository. So this is a very simple way how you can start your work. And once you measure something, you can use Aquaexcel repository to store the data and link your data with this Aquaexcel TNA form protocol. And later on, you'll be able to visualize all your data measurement and processing steps in the web interface, including the application form. So this is a very simple way how you can use the Aquaexcel repository for the TNA applications. Now we are at the end of the training. So during the 3 days, I try to show you how and what you can do with the system. Hopefully, you were able to create a protocol template and protocol. If you will have any problems, you can still contact us, and we will try to help you. All these 3 sessions were recorded, and the records will be available online next week. So we will send you the link to the video records. So if you missed something, then you can simply play the videos and you can find out how to solve your problem, or you can still contact us. Another thing is that for the participant, we prepared the certificates. So again, next week we will send it to you. It will be mentioned that you were introduced into the system of the data management Aquaexcel repository, and we will kindly ask you to provide us the feedback from the course. So we will send you the link to do a questionnaire with a few simple questions about the course, about the understandability of the training, and so on. So please try to find some time and just fill these very simple questions to have the feedback to improve the training for the next time. So thank you very much. Again, we will be sitting on the chat. If you have any questions or problems, then we will still try to help you. And thank you very much for the participation on the training, and hopefully it was useful for you. If you decide to use Aquaexcel repository for your experiments, then feel free to use your already created account. Or if someone see that the repository is useful and would like to have the repository directly in the lab, it is not a problem to contact us, and we can try to prepare the repository for you, for your lab, so it will be fully under your control. Aquaexcel repository, it means the server, the storage itself, it's located here in our institute, and we have the full control over that. But don't be scared, we don't have the access to your data, so the only person who has the access is you, and if you share the data to someone else. So thank you very much.